So, okay, so if you're diagnosed with sporadic MND, can you pass it on to your siblings? It depends on what so the answer is traditionally no, but that gene that I told you about, CREST, is an example where there's been what we call a de novo mutation in one of the parents, but it was only in the germ cells, it wasn't in the body. So that means that parent isn't subject to the negative effects of that mutation, but they will transmit it. So I think the barrier between sporadic and inherited MND is starting to become very blurred. I think there is an increasing view in the research circles that sporadic MND may be potentially mostly, if not entirely, genetic. And I think this is good news from a therapeutic standpoint. So I think the answer to your question is yes.
We're just waiting for the microphone further.
What is your opinion of if your MND is caused by the C9 gene, that the younger generation being tested, is there any advantage in this, knowing that it may not, if you do have the gene, it may not manifest itself?
So that's a good question. So C9 is quite similar to Huntington's disease gene. So the problem is that with each generation, the genetic stutter gets larger, and then you end up with a phenomenon known as clinical anticipation, where you get an earlier onset of the gene. I just wanted to point out that the diagnosis of an MND gene using a genetic screen isn't an instant death sentence. So there's a very good case where there are two sisters who both carry a SOD1 gene mutation, yet only one of them developed MND. So I think that is a really, it's certainly an ethical quandary as to what you do with that information. However, if there was an anti-C9 therapy and you had the ability to diagnose the disease before it manifested, that would be, one could potentially avail themselves to that therapy.
Arlene.
Sure.
Thank you. I think you might have implied the answer to my question, which is, does the number of repetitions in the CR9 mutation increase the likelihood of manifesting motor neurone disease?
Yes.
And that's going to increase with each generation?
That's right. So there is a generational increase in the genetic stutter. The other unique thing about C9 is that it has what we call somatic instability. So the repeats can vary depending on which tissue or cell that you're in. So that adds another element of complexity to the heritability of a C9.
I'm not.
What screening processes do you go through to determine what patients will be suitable for clinical trials?
That's a good question. So, for instance, that anti-SOD1 clinical trial only enrolled, obviously, SOD1 positive patients. So there's now a belief that in some circles people have, they're beginning to relent with testing drugs on sporadic MND, and instead they're focusing on those patients with specific gene mutations, which will be a prerequisite to be enrolled. However, I just wanted to add to that. I showed you a slide before where abnormal TDP43 or SOD1 or FUS accumulates in the brain or in the motor neurons of sporadic MND patients. And this is why I think these results from the anti-SOD1 or the anti-C9 therapy are exciting because they're also applicable to all forms of MND. This is what's so encouraging about these clinical trials.
Brad, I was thinking about most of the research has a very genomic focus.
Yes.
Whereas the problem is really in the phenotype as is expressed. So I'm thinking about biomarkers and proteomic research that could introduce maybe a biomarker or early indicator that people are developing MND.
Yeah.
Is there much crossover between the proteomic people and the genomic people?
There is. So there's very active biomarker programs in Philadelphia, in Oxford. There's actually some candidate biomarkers, so that I think they've identified 3 or 4. There's certainly a lot of crosstalk between, ultimately it's the proteome and the biomarker profile which matters and is important for this early diagnosis potential.
Questions?
With regard to C9 specifically, is this gene only inherited? So say, for example, if the parent is negative to C9, is there a chance, other than a sporadic form, that the children can have C9 if the parent has failed the C9 test?
Yes, I think C9 it's found in 5 to 8% of sporadic MND patients. So that means the parent did not have the expansion, so it can arise spontaneously. The other thing I'll point out about C9 is I tend not to think of C9 as a single genetic cause of MND. I think it is potentially a risk factor and a modifier, and there's been a change in the MND genetics, the way we think about it. So we now think of this oligogenetic inheritance where you have potentially 2 or more genes interacting in concert with lifestyle and aging factors. So it can arise spontaneously.
Any of these genes are connected to people with inherited heart disease, genetic heart disease as well?
Not that I'm aware of. Most of them are linked to, for instance, Alzheimer's disease shows some overlap with TDP43 and C9, and that's really getting at this risk factor to MND.
Any other questions?
Is there any preventative measures that can be taken?
In terms of prophylaxis or prevention, no, because again, MND is largely sporadic, so 90% of cases are sporadic, and of those we've only solved 12% genetically, so unknown currently.
Can I ask a question? Can anyone just go and get their genes tested to see if they have a MND gene?
I believe that's usually a referral by the neurologist. So yeah, I've actually asked. So Ian Blair, I actually asked him once how long would it take to sequence my genome. Would he volunteer to do it? So he said it would take about a week. But usually it's by referral, and it's usually what we call a candidate gene response. There's a panel of the known MND genes which they will sequence first.
My question was, sorry, can anyone just go and get a test to see if they have the MND gene?
So I would only do that if you have a diagnosis from a neurologist, or if there's a family history of muscle wasting or MND-like